We'll get right to our next presenter, Ashley Elgin from NOAA GLERL up at the Lake Michigan Lab in Muskegon, talking about invasive mussels. And Ashley, take it away.
Thank you very much. Hi, everyone. Yeah, so I'm Dr. Ashley Elgin. I'm a researcher based at our field station in Muskegon. NOAA GLERL has a Lake Michigan field station there right at the Pere Marquette Beach next to the Coast Guard. And today I'm going to be talking about invasive mussels, quagga and zebra mussels, and where we're at for control options and tool development. I just like to start out in the beginning. I'm fortunate to work with a lot of great people in the course of the studies. A lot of people contributed in some way or another to the figures I'm going to show. So I just like to thank everyone up front and recognize it's many academic and state and federal agencies that contribute to this kind of project. So what I'm going to talk about is Dreissenid mussel background. Some of you may be very familiar with Dreissenid mussels. It might be newer to some of you. Tell you about the Lake Michigan sampling that we do, what's happening with the population trends with our most recent data, talk a little more about their body condition and size distribution, because I thought it's a fish group. You want to know about body conditions, see what's happening with sizes, and then also talk about the veligers. That's their larval dispersal stage, and that's what's happening with larval, what's up in the water column for the mussel veligers, and then finish up on mussel control. And so getting here today, I got here a little bit early, so of course I had to go to the beach. Walk down, I get to enjoy, get to see Lake Michigan, say hello to some of my sites that are offshore there. B3, hi, how's it going? I get to marvel at the amount of mussel shells that are in the sand. So of course, I just can't help it. I took some with me. So you see that we have, many of you, if you go to the beach, you see piles of shells on the shore. It's just a glimpse of what's offshore in the water. And that's what I hope to show you in data today. So I'll be talking about two species. I use Dreissenid, that's the genus, and it's the quagga mussel and zebra mussel. Who is more familiar with zebra mussels than quagga mussels? All right, who knows all about quagga mussels too? Then that's good. This is I hope to let you know that both species are very important, but when you're out in Lake Michigan, it's a quagga mussel story and has been for a while. So the impacts of Dreissenid mussels, they reduce the spring diatom bloom because they are amazing filter feeders. I'll show you a picture later of they just have siphons that go out. Every liter of water that's in Lake Michigan could pass through a mussel in a week's time because there are that many mussels and they filter that actively. So in that, they reduce the beneficial spring diatom bloom. By increasing water clarity, you can get more nuisance algae on the bottom, the Cladophora and other benthic algae. In that way, they shift energy from the water column down to the bottom of the lake or the benthic zone. This picture here shows that they will foul or cover native mussels and smother them, and they can displace native mussels. And then you've probably seen a lot of this. They foul infrastructure, they'll muck up your pop, they'll get all over your dock, they clog water pipes. So they cause a lot of issues. They also can cause changes to the benthic or the bottom dwelling invertebrate community. So this graph, I'll step through it slowly, and I know this is tiny. This is a very cozy screen for all of us. So I'll try to just step through that. It's showing over time, starting in 1980, well before the mussels arrived. The lake was dominated by Diparia, which is a very high quality food source of bottom dwelling amphipod or scud. And then also oligochaetes in blue were present, and then native small fingernail clams. So this pie chart represents what was happening in 1980. So mostly Diparia in green. Then you go over time at the star there at this point, mussels arrived and things started changing in the lake. The Diparia have been on they were on a steady decline ever since then, so that as of 2015, we have very few Diparia in the lake. The yellow portion is what's exploded. These are the invasive mussels. And then oligochaetes are still present. They're doing similarly well in about the same proportion. But you can see you've gone from a Diparia-dominated bottom environment to a mussel-dominated environment. And that means if, say, you're a lake whitefish, you're foraging on the bottom, your dinner plate looks quite different. And the whitefish have responded, they have lower body condition. They don't thrive on this diet the way they did on that diet. So I'll spend a little time on biology because I'm a mussel biologist. I think about these species all the time, and I have an opportunity to talk more about them in more detail. I'll go over this quickly, but if you have more questions, I'm happy to come back to it. The main thing is that this two species, they're like cousins. They're very closely related, but they have different preferences. So zebra mussels are more in hard surfaces. They like warmer temperatures and shallow depths. So zebra mussels are what you'll see in nearshore inland lakes. That's a zebra territory. But quaggas, they can anchor in soft substrates. They can handle lower temperatures, and you'll find them shallow to deep. So I'll be showing you some data very quickly here about how lakes like Lake Michigan and deep cold lakes are very suitable habitat for quaggas. As far as distributions go, quaggas are dominant in the Great Lakes. We rarely find zebra mussels offshore at all. The zebra mussels, as I said before, are more common in inland lakes, infrastructure, and in rivers. And if they are going to coexist, it's going to be in these shallow, productive areas like Saginaw Bay, Green Bay, and Western Lake Erie. So in order to learn about this highly impactful species, the lakes, there's the lakes before mussels and the lakes after. And NOAA has done a lot of research to track the populations of these mussels over time. The approach is annual surveys in the southern portion of Lake Michigan. I'll be showing that data today, but there are also whole lake surveys on a 5-year cycle. And we have many cooperators and partners who've been part of those whole lake surveys over time. But I'd like to focus on this annual benthic survey. It's wonderful to have annual data. Imagine if you have data that's every 5 years, a lot can happen in 5 years. A population can zip up and crash down within 5 years, as I'll be showing you soon. And so it's valuable that we just focus on the southern region of Lake Michigan. We look at it every year, and we've been doing that since these surveys, as it is now, were established in 1980. Some of the sites go back to the 1960s, and we've been doing it annually since the 1990s. The original goal is, the phosphorus inputs were reduced going into the lake, and they wanted to see how are the bottom organisms responding to this reduced phosphorus. But then Dreissenid mussels were introduced, and this survey became an opportunity to have baseline data before and then see what's there after. We also have an opportunity to see how this invasion developed over time. So I'm going to step you through a series of plots, and if it's darker blue, that means there are more mussels in that region. The top is zebra mussels, and then the bottom is quagga mussels. So in the very beginning, in the mid-90s, everyone's hearing about zebra mussels. They're exploding everywhere. You're going to be seeing them in nearshore areas, and there are no quagga mussels yet. And then in the next 10 years, you have zebra mussels. They're successful. They've a ring around the entire lake. They managed to get to every area. They're in Green Bay. And the quagga mussels in 2000 are just showing up in the north. But then what a difference from 2000 to 2005 in the quagga mussels. They flashed all around the lake, and they went deeper than zebra mussels were ever found. Meanwhile, zebra mussels, their populations are stepping back. They're waning. They're getting displaced. Now, put another 10 years of data on there. In this survey, we don't even see zebra mussels anymore. Quagga mussels, they can do deep, they can do cold, they can do soft substrate. They colonize the entire basin of the lake. And then 2015, it looks very similar. I don't have the 2021 figure on here, but Buffalo State College or Buffalo State University produced those data. And the map looks very similar to 2015. So they're very established throughout the lake. So seeing it as a density map is one thing. I'll show you a picture of what does this density level look like. And I'm going to jump ahead because I can't wait to show you. That's what it looks like when we talk about that type of density. I don't know if you could walk across the bottom of Lake Michigan on a mussel, but you could get pretty close. So in the very deep areas, they might be more sparse, but this is at 45 meters depth. This is the equipment we use to sample. That's a Ponar grab going down. A documentary company, a documentary team went out with us, and they had an ROV down, or underwater vehicle there, to film our sampling, and we got this great shot. And I love it because it shows, it's the only way to really see how dense the mussels are down there. So now I'll step back to, I showed you these, this is a big picture. This tells the quagga and the zebra story very nicely in these plots. Now I'm going to show you our Southern Lake Michigan data. It has the benefit of catching the changes from year to year and what it's telling us. So to start out, I'm going to bring in the depth zones one by one. The gray line is the most shallow zone, and the numbers are bouncing up and down. These error bars, the bigger the error bars, it means there's a lot of variation from one site to the next, because the nearshore zone is a high energy, the sand is shifting, you can have a storm that could smother a population. So you just get a lot of bouncing around in their numbers. And that's reflected right here in the data. But then you get a little further offshore, 30 to 50 meters. This is where annual data is really helpful, because you could see just in the first 7 years of the invasion, they raced up to their peak and they had higher numbers that were ever recorded. And then over the next 2 years, they crashed back down to less than half of that. And this is what's informative, is ever since then, they've been bouncing around. So you think that they overshot what the lake could carry, and then they came back down, and they've been bouncing at this level around 20 grams of ash-free dry weight per meter squared, maybe a common measure for everyone. It's just a common way that we use to measure biomass of the mussels. So now let's keep going further offshore. And we have this next depth zone of 51 to 90 meters. And also it jumped up and then it came down below the peak, just not as dramatic. And then next, this is our deepest zone. And a majority of the lake is in this deepest zone. And it was a slow and steady increase to what appears to be kind of this bouncing around. That's what the lake at that depth can support as far as mussel biomass goes. And then we get to see this picture again. Oh, wait, first, I did a weighted mean, weighting by the area in each depth zone. And you could see that the overall weighted mean, again, is staying pretty steady after a really rocky and flashy first 10 years of the invasion. Just really, it's leveled out. And in this way, we can see with this finer annual data that the populations are more stable at this point in Lake Michigan. So this picture, again, it blows me away every time. Another way that I look at mussels is not just their straight populations, but I take the body condition of the mussels, which indicates their nutritional state, where they are at, their reproductive status. And these are all the sites that we've sampled since 2015 across all of the lower Great Lakes. We don't do Lake Superior because there aren't enough mussels up there to do this analysis. And so in Lake Michigan, I've had the sites circled here. We've done a lot of these sites twice so far. And every year we'll do one of the lakes in a rotating cycle, and we make all the data available for anyone who's interested. There's a link here on the slide. So the main takeaways here, we took all these length weight regressions. So given, in a given length of a mussel, how much body tissue is there? And we're putting this in terms of how much tissue is there in a 15 millimeter mussel, pretty average size. And what we find here is in the depth zones, there are significant differences among the depth. And in each of these depth zones, this is shallow going deeper. Each of these pairs is the 2015 in the dark bar and the 2021 in the light bar. And there are significant drops in their body condition going over that 6-year period. So mussels in the near shore, just to sum up, have the heaviest mussels for a given length.
Based just on nutrition, like as far as chlorophyll, like the diaphragm, the freshwater shrimp, were they also, is that what was their nutrition? I mean, filtering the shrimp, I mean, is that what they were feeding off of to actually grow and actually feed? Was that, did you see correlation with our freshwater shrimp and the mussels? Is that?
So in general, we've seen that as the mussels have come in, the dipraia, they've gone down. They eat similar food. So the mussels are filtering the phytoplankton, small algae right out of the water, and that's their primary food source. And there's it's been hard to make a strong connection between the presence of mussels coincides with the drop of dipraia, but it's been hard to make a definitive conclusion if this caused that.
Are you finding more still in the southern part of Lake Michigan as far as, you know, the freshwater shrimp, the dipraia, or are they?
Actually, more in the central and the north. So we have some sites that were this deep site in the middle of the southern basin. And in the last 4 years, we haven't found one there. And that was like our holdout site that we still had them in the south. So we've been seeing the dipraia to continue to go down. They show up here and there, so they're not gone. They're just kind of below our detection limit, and we find them in fish diets.
I've seen that, I've
'Cause-
seen
yeah.
less than our total.
You have, okay, ever?
I still see some.
Mhm.
I check every stomach, and that used to be their main diet.
Okay, so you saw it, yeah.
Absolutely,
Mhm.
absolutely, and
Mhm.
I haven't seen that.
Yeah. So they've really gone down the south. There are pockets in the central and north part of the lake where you still find them, but way below what they were before. This is using some more local data, still looking at body condition, but this is now the Muskegon transect. We look at this body condition metric of mussels every season. I just finished analyzing some of these samples this morning from our spring time point. And it just shows that M25 is the shallow one. Those are the beefiest mussels that bounces around over time. And it hasn't shown much of a trend, but they are the warmest temperatures. They're the best fed because they're near shore. But the main thing I want to show you is at the offshore mussels in these two lines, the green and the blue, they've steadily gone down over time. So the environment there was more food to support mussels when they first arrived. The mussels were heavier, their populations exploded. And since then, the mussels themselves are removing food, and it's less food available for the mussels. So we've seen that shift over time.
I got a question.
Mhm.
With the mussels, you doing this, doing that, is that why we have no smell here?
I'd look to Dan on that one. Yeah, so I mean, they're clearing out the base of the food web. So they're few, if that's less food for smelt, less food for alewife, that could be having an impact, but it's really hard to pin that down exactly. Yeah, but that's a mechanism, and I believe that is happening. If that's the only reason, I can't say.
So what's prey primarily in mussels, especially the
Yeah, so, the veligers, well, I'll start with the standing stock, the adult mussels on the bottom. Round goby are really good. Round goby come from the same region as mussels do. They have the same native region. So round goby get over here and they're like, hey, I know you. They eat them up. And then whitefish also will eat mussels, drum eat mussels. There are a few fish that will use them as a food source. As far as the veligers, there are some larval, because they're so small, larval fish will eat them. Yellow perch will eat the veligers. Some fish appear to actively avoid the veligers. And I can mention a little more about that when we get to the veligers at the end. So here, I just want to mention about, the body condition is one way to learn more about what's changing with the mussels and their environment over time. And then that could help us understand the population changes. Another thing is I like to look at the size structure, and this is a simplified figure in that this is just the mussels less than 5 millimeters, indicating what are the mussels, kind of think of as the young of the year class that have just recently came in. And yeah, sorry, this is covering, but it's showing shallow as you get deeper, and the color of bars just shows over time, over the last 6 years, what's happening with size structure. And in general, you have fewer small mussels now than we did before. And those are fewer small mussels to fuel population growth going forward. I did this figure to zoom in because they were small, but you can just see a general decline over time, that you have fewer small mussels at each of the depth zones. I did want to give you some South Haven. This is some regional specific data. This is this would be called the South Haven transect that just goes offshore here. I have data just for that transect, just to show you what's happening. These are, this is overall biomass. And again, the color bars is going over the last 6 years of data that we have. So near shore, density can be really low against a volatile, shifting sands, high energy environment, and that can, it's not the best for mussels. But then you get further offshore, and that's where you have the highest biomass. And then getting to the deepest sites, we tend to have a little lower biomass. So this is what's happening with mussels in your neighborhood. I just wanted to show you those sites. So just a few words about veligers, then I'll get to control, wrap up here. So veligers, they are, so mussels are broadcast spawners, external fertilization, sperm and egg go out, they fertilize and they make these veligers that float around in the water. These veligers then will spend a good amount of time in the water. They're there as a food source for fish. The veligers are eating phytoplankton while they're in the water, and they will drift on the currents and they will disperse. And that's how they were able to get around the lake within 5 years' time, is because they have this larval planktonic dispersal stage. So that's a really important part of why they're so successful for as invasive species. Yes.
They move vertically, so I know.
They do. And they can move vertically. And then when we sample them, they tend to be concentrated in the upper level of the water. And if they weren't actively moving, that wouldn't be the case. But we find them throughout the water column, but they prefer to be up high. This data, this is also from our Muskegon transect. These are just days of the year, Julian date of the year. What I want you to see here is there are 2 peaks. So it shows it evidence, and these 2 peaks are happening at these 3 sites simultaneously. And it just shows a synchronized spawning event that you get 2 per year. The other thing you can see with the size distribution, you have a peak here where the size is small. Those mussels get large. They get large enough, they settle out of the water. And then the average size goes down again because you have a new class coming in, a new spawning event. And that's later in the season. So you're kind of looking at a late June and September spawning. So just to show some of we're taking a closer look at veligers, because, the veligers of today, small percentage of them survive, but those are the mussels of tomorrow. And then also look at from year to year, you can have a lot of variation. It almost looks like a cycle. You have kind of boom, it gets a little low, you have high veliger years. And this last year is 2021 shown here. That's a low year. And so even though we're seeing these clear patterns, in the scheme of things, it's pretty low veliger production. But then this figure has data up through 2023 at the nearshore site. I think they had close to, it was a sample that had almost 1 million veligers in it. They don't count them all. They just count a portion, but it comes up to being almost 1 million veligers. It's one of our highest samples we've ever seen. So 2023 is shaping up, at least at this, at the nearshore, produced a high level of veligers. So what will that mean? I'm keeping my eye on, what does that mean for the populations, in the next couple of years? Because it's a big class coming up. Mm-hmm.
How long do they live?
Oh, it depends on the temperature. So in the near shore, they might just live 2-3 years. Like think of Lake Erie, it's warm. They'll hit their maximum size and live with just a couple of years. Offshore, I've seen evidence that they could be living for a decade, even more, because they'll grow slow and they live long in the deep cold water.
You found any way to kill them?
Yeah, well, Goby help. Goby are a good way to kill them, but I'll be moving. Yeah, I think my next slide is what can be done. So thanks for keying that up. I'll give you your $5 later. So this picture here, this is a close-up of the sea of mussels. Their siphons are out. You can see them extended. I know it's hard, but they have all these siphons, and that's just them constantly feeding. I've watched video of them around the clock as a time lapse, and they rarely stop feeding. They're just always feeding. So this is how they have these huge ecosystem effects. So a year ago, the Invasive Mussel Collaborative formed, and it's the whole point, or 10 years ago, is to advance scientifically sound technology for controlling mussels. One of the things that led it was a development of a new toxicant I'll talk about next. There are several toxicants and physical treatments that are being developed and examined, and the website's getting covered up here, but it's invasivemusselcollaborative.net. So one toxicant that it made a lot of news is called Zequinox. It's Pseudomonas fluorescens. It's a bacteria that's found in soil. And they found that this bacteria will, if you feed it to a native mussel and you feed it to a Dreissenid mussel, it's only lethal for the Dreissenid mussel. It gets into their digestive tract. It causes their digestive tract to die essentially, and they'll die in a few weeks. So that is, if you have a native mussel and it's covered with invasive mussels, that would be a toxicant you would use to be very specific. But then there are other chemicals such as potash and the closamide. This is a lampricide that's used to treat sea lamprey, and then copper compounds. These are less specific. Those are generally very toxic to invertebrates and vertebrates as well. And so those testing out, you want to use the lowest concentration possible. Other ways, if you don't want to deal with chemicals, you can use benthic mats. They can be laid down to smother the mussels. This is a treatment that's happening in a Texas lake. They just put it down. They consume all the oxygen, the mussels smother, and they suffocate. USGS is doing research to see, can you put carbon dioxide, even small amounts of carbon dioxide under that tarp to speed up the process, and it's lethal for them. In lakes that have water control structures, you could do drawdowns to kill nearshore populations. And then there's also physical and manual removal, which you can imagine is time consuming and involved, but can be highly effective. So I'm involved with the project, and I have collaborators from the University of Wisconsin-Milwaukee. And our goal is to test these underwater vehicles or implements for destroying mussels. And the ultimate goal is, I know it's small scale to begin with, but you have to start somewhere. And it's to see what happens to nutrient cycling, what happens to the other bottom dwelling organisms when you try each of these methods. And then you have to learn that, and then can we scale it up? So here's a picture of what's called the mussel crusher. They did this over in Milwaukee, and they pulled it behind a boat, and you can see this path of destruction from where this crusher was brought through one of those mussel beds. You know, this mussel bed picture might look familiar from the picture you saw before, and that's their path of destruction. And they'll continue to monitor how long or how quickly does it take the mussels to recolonize? What's happening with other benthic organisms in that path? And then on our side, around Muskegon, we're planning for an underwater vehicle test to have a tracked, it would be autonomously running tracked vehicle that would run on the bottom. And right now we're designing that it would take up the sediment, maybe crush and sieve out the mussels, put them through a roller, and just leave the mussels behind. And then, of course, what happens with the decaying mussels? What does that do to the nutrients? How do the invertebrates and the other benthic dwelling organisms respond to that? So those are things that we're just in the beginning years of this project.
Is that practical given the size of the lake? Option?
That's a good question. So one thing is, if you can have, so if you picture like a mat, something, you can have that mat, and then you have to get in, you have to move it to the next spot and move it to the next spot. But so that's scalable, but it's a lot of effort to move that treatment around. If you could have something like a vehicle, imagine if it had an energy source you could even have underwater, it could just be around the clock, running out there, doing its thing like a Roomba, and it would go through. And, you know, in that way, it is scalable, but it would be gradual. Take a little time. They can operate at deep depths. That's what's nice about some of these. But again, this is we're just at the beginning of starting to test it out. Your skepticism is very valid. This just shows the logistical challenges of doing something on such a large scale. So, yeah. But just think of that. There's a sea lamprey program. You know, they spent decades developing that, and they came up with a basin-wide solution that saved Great Lakes fisheries. So there's always somewhere you have to start, but all of these are wonderful points. There was a Zequinox field test in 2019 at Good Harbor Reef, and it was very successful. They found 90% mortality a month later. And then additionally, they found round goby were very effective because they eat small mussels and they target small mussels in slowing down the recolonization of mussels in treated areas. And this is work with the National Park Service and University of Milwaukee Wisconsin has been involved in doing work here, and many other partners. So based on the success that was found with this test, a field test, there has been a federal working group for experimental muscle control that's been formed. And the idea here is to have Great Lakes Restoration Initiative to have funding for this project to focus on a habitat of very high quality and high value. And that would be spawning reefs for lake whitefish production. The hypothesis is if you could reduce mussels on the spawning reef, can you improve the whitefish production by increasing the embryo survival? Because if mussels are filling up all of the interstitial space on this reef, is that reducing, is that another reason for the declining whitefish recruitment? So this project it's a 5-year project. It's just beginning now. It's going to be multiple stages. Right now it's selecting sites. The lake technical committees for Michigan and Huron chimed in, and we found some top sites to try this out, good candidate sites. And it's a collaborative effort, multiple agency, tribal, and academic partners. And we're in the beginning stages of it. So stay tuned. I hope we have some successes to report.
Has there ever been any work done on DNA mapping to find some flaw in the genome that they could alter and keep them from
Yeah.
reproducing?
So there are ongoing genetic tools being developed. That is, yeah, there's gene drive technologies, a way you could alter the genome. There's work to try to find, is there a disease that we could use to kill the mussels? And that's something that would be, it would be catastrophic and rapid to the populations. That itself would be a shock to the lake. I just want to put that out there. They've come to the point where they are fairly gradually. So if there's a massive die-off, that would be, the lakes would be reeling from a massive die-off of mussels too. So if we got to that level of technology, it would have to be deployed in a very careful and controlled way to the extent we could.
How the life cycle goes in the source areas for these mussels, where they came from, and whether there's anything we learn from that to control mussels.
Well, in the native range, there are going to be more fish predators that control the mussels. And in the native range, they're not found in the deep, large lakes like they are in the Great Lakes. It's actually quite unique that they came over and do so well in the Great Lakes, because they don't have an analogous they don't have a Lake Michigan where they came from. It's a different environment. It's more rivers and smaller lakes. So as far as what we would learn from the native range, there are researchers trying to find, say, a native parasite or something like that could be used for control. And that's another area of research. So I think I've spent plenty of time here. I don't know if there's a question online. This is my email. So please jot this down. Reach out to me anytime if you have follow-up. So the way I say shock to the lake, because if you were to have a massive die-off of mussels, that would be a huge amount of, the biomass of mussels is staggering. And so then if you were to have that decaying, it could consume oxygen, it could cause hypoxic zones, it could cause die-offs, it could throw off the nutrient cycling in the lakes. And so any abrupt change in the mussels, that's why I say it would be a big shock to the system.
So most of this research done, are you going from boat to boat, or do you do a lot of it on the same vessel with the university?
Yeah. So a lot of it, so right now this picture is me standing because the field station's behind me coming in the Muskegon Channel. This is our 80-foot research vessel, the Laurentian. I do a lot of work on that. But the whole lake work, that's done on the EPA research vessel called the Lake Guardian. So it's a lot of different boats. For NOAA, we have a fleet. There are about 3 of the different NOAA boats that I use commonly. It just depends on what region we're working in. Yeah.
Where does your funding come from? And are you getting enough to do this, in your opinion, adequately and quickly enough to satisfy the public?
It's a great question. That's a big question. I'll give a lot of credit. So the Great Lakes Restoration Initiative, our lab we've done extra projects. We've done a lot of additional work that we've only been able to do because of Great Lakes Restoration Initiative funding. Otherwise, we have base funding from our lab, and that's just what's given to us from Congress to do a lot of our monitoring work, because that's something that we do over time.
Is the tribe giving you any money?
We don't have any, well, that's not, most of the money that we have comes from our own agency or GLRI. So that's usually how the funding flow goes for us.
But what would you say about adequacy?
Adequacy.
I mean, I know you'd always like more,
Yeah.
but
If I could clone myself, or if we had more, I think at this point that it's more of not having enough researchers who can do it, because we've had a really good funding, but the capacity to run a research program and the list of the questions I would love to do and the studies I'd love to do, but I'm like, I need more, more of us to do it. And then that comes down to funding. But for those of us who are available, we've been in a good time for research, and we've been able to answer there's still a lot of unknowns, and we've been able to do it, but there's always more to do. So with that, we need to keep getting more people in the pipeline. We need more scientists. We need more science. So, all right. Thanks everyone for your questions. They're great. And yeah, thank you very much.